Can everyone hear me? Okay. So welcome to our very first EEB seminar this year. I'm very happy to introduce Jennifer Powers, who, despite all the craziness with COVID and flight cancellation, she finally made it and is here in person. She did her PhD at the Duke University, and then she finished her postdoctoral work at Stony Brook and at the University of Minnesota. She has been an adjunct professor of the Universidad of Costa Rica for a few years. She's also a research associate at the Smithsonian Tropical Research Institute, and since 2006, she has been a professor at the University of Minnesota in the EEB department and plant biology there. And her work focuses on understanding how the functions and structure and the function and structure of tropical dry forest, going from local to ecosystem levels. And today she's going to tell us a little bit about her work in this system. So with that, I will just let you talk. Welcome, Jennifer.
Thank you. Yeah. Thank you so much for the invitation, Natalia. Let's see here. Should I wear this? Or should I wear this? Yes. Okay, thank you. Yes. I feel like this is the first time. Okay, let's see. This is the first time I've given a talk in person since the pandemic, so I'm just as out of practice as you might be. So anyway, you guys can laugh. You don't have to. I can't see under the masks. Okay. Okay, can you hear me? Yes. Okay, cool. Awesome. Well, thank you. Like I said, thank you for this invitation. It's really great to be here in person. I think there's a lot of crazy stuff going on in the world the last couple years, and it's just such a delight to be back talking science with awesome people in person. And so I think of you guys as the other U of M. You probably think of us as the other U of M. But anyway, it's just, for me, it's a delight to be here. So thank you. So for the next hour or so, we're going to talk about the bosque seco, or tropical dry forest. And so this, I think, was something along the title that I suggested, something vague enough I can kind of incorporate lots of different things into. So I think that all of us here may have some understanding, or at least some mental image, of what a tropical rainforest looks like, even if you haven't been there yet. You have some idea of what it looks like. By contrast, there are many people who are surprised to learn that not all tropical forests are tropical rainforests. And in fact, all of these forests here would be categorized or classified as tropical dry forests. And tropical dry forests as a biome used to once account for maybe 40% of all tropical forests would have been classified as tropical dry forests. So the question I have here is, what do we know about tropical dry forests? Well, we do know some things. We know that they have high biodiversity, and the biodiversity that we find there is often endemic. We also know that they have high functional diversity. This is a slide taken in the dry season in Área de Conservación Guanacaste, and you can see in those circles that there's lots of different types of trees. Here, we have deciduous trees in yellow, co-occurring with evergreen trees in green, and semi-deciduous or brevideciduous trees, which you see there in blue. So there's high functional diversity. We also know that this is an important biome for supporting livelihoods. Tropical dry forests provide many ecosystem services, including land for agriculture, tourism, provisioning of water, and fuel wood. We also know that these are highly threatened ecosystems. They have been converted to lands like agriculture. They suffer from global changes, including things like drought. This is a picture here. I've talked to a number of people about invasive species.
They are threatened by invasive species like the tree in the, I guess, northeast quadrant there. And they're also threatened by drivers like fire, just to name a few. So now I want to focus more on what we don't know about tropical dry forest, which turns out is like pretty much everything else, I guess. So the goal of our lab is to understand the patterns and the mechanisms through which seasonally dry tropical forests respond to changes in land use and to changes in climate from microbial to biome-wide scales. So that's our goal, and we use a number of different conceptual approaches or conceptual lenses to achieve this goal, including community assembly, functional ecology, and ecosystems ecology. And to achieve these goals, we use a number of different approaches or tools, like data synthesis, large experiments, long-term observations, and measurements of traits. So what I'm going to do today is just sort of walk you through some of the work we've been working on over the last, I can't say over the last 2 years, because definitely our research has taken a detour over the last 2 years, but over the last 10 years or so. And these are the topics that I want to address. Thinking about the structure of tropical dry forests, and in particular, in the Americas, one plant family that is very abundant, and that is the legumes. And then I want to focus more on some of the functions, or the functional processes we can observe in dry forests, including how they relate to drought, or how they're responding to drought, and which traits mediate these processes. And then for the remainder of the talk, I'm going to kind of shift focus and think about how we can consider tropical dry forest in a broader, more geographically broad, I guess, context. And think in particular about hydraulic traits and also soil biogeochemistry. And last, if there's time, I'm going to finish up, if you guys are still awake, and who knows with masks on, right? I'll finish up with a bedtime story. Okay, so this is an outline of the talk in pictures. So we're going to start by thinking about legumes. Here in the Midwest, we think of legumes as soybeans. We think of them as herbaceous plants. In the tropics, you think of them as large trees. Trees from the family Fabaceae, or the legume family, Leguminosae, or the pea family, they comprise a large amount of the basal area and the abundance of trees in the study site where we do most of our work in Guanacaste, Costa Rica. So this is just the percentage of trees in 8.4 hectares that we've inventoried. And you can see that Fabaceae account for about 18% of the abundance of trees in this landscape. And so that's interesting from an ecological perspective. From a biogeochemical perspective, we know that Fabaceae are important because many of the 19,000 or so species can fix nitrogen in symbiosis with rhizobia in root nodules. What has been lacking, however, is sort of a continental scale understanding of the patterns and the drivers of legume abundance, certainly in the neotropics. So this project was led by a postdoc in my lab, Dr. Maga Hay. And what we did was to understand the core scale constraints on legume abundances. We collated a number of chronosequences. So these are pairs of plots that differ in forest age, and each one of these dots here represents a chronosequence, and these chronosequences were arrayed along a climate gradient. So what we really wanted to do was understand the core scale controls or patterns of legume abundance across these different gradients of secondary forest succession and climate, or rainfall. So this plot here summarizes literally tens of thousands of trees. Each one of those dots is a forest inventory plot The vertical axis shows the absolute basal area, and the horizontal axis plots that as a function of stand age, or forest age, for these secondary forests. Each one of those lines, then, is a single chronosequence that is represented by those different plots. And we categorize these chronosequences based on the rainfall regime into dry forest with a threshold of about 1,500 millimeters of rainfall, of annual rainfall, to wet forest, which we categorize as forests having over 1.5 meters of rainfall. And so dry forest in orange, wet forest in blue. And what you can see from this, the mean values for dry and wet forests are in the inset above. You can see that, kind of reassuringly, the basal area of trees increases through succession. That tells us that, succession is working. But you can also see that there's comparatively more basal area of legumes at any given successional age. So in this plot, what we've done is look at the relative basal area of legumes. So it's the same data set, but basal area is expressed now relative to the total basal area of the plot. And so what we can see here in the inset, is that if we really want to understand where legumes are abundant in neotropical or in forests in the Americas, they are most abundant in dry forests, and in particular in early successional dry forests. So for the first time, this study really revealed these sort of continental scale patterns of legume abundance in the Americas. But these patterns don't tell us much about the mechanisms that account for them. So we say that legumes are not just different, they're better. They just seem to be better in these ecosystems. And we know a lot of things that might suggest why they're better. And these factors here are not mutually exclusive. So we know that they have high foliar nitrogen in general. And part of this is is correlated with their ability to fix nitrogen. We know that at least the legumes that we've looked at for the neotropics have small leaflets, and this might give them an advantage in arid climates. Other people, like I think Mark Adams, have collated data that suggests that legumes have high water use efficiency compared to non-legumes. And again, none of these mechanisms are mutually exclusive, but we're advancing another hypothesis for the place we work in Costa Rica, and that is the hypothesis that legumes have a different regeneration niche. So now what we're going to do is sort of drill down into this study area in Costa Rica, where I've spent the last 20 years of my life working in early to mid to late successional forests in the Área de Conservación Guanacaste, or Santa Rosa National Park. So we're going to drill down into this one region in Costa Rica that has a lot of legumes. And Costa Rica is awesome, by the way. You guys should all visit. So what we've done is to do very, very simple experiments in shade houses to examine this hypothesis that legumes might have a different early or regeneration niche, a different niche at the early stages. So in this experiment, what we did, again, super simple. We took 9 species of legumes, and we compared them to 23 species of diverse non-legumes. And this is super simple. What we did was to apply 2 different germination treatments to all of the different seeds. We either immerse them in boiling water for a minute to try to break the seed coat and then put them in cold water, or we just soaked them in water overnight. And here you can see cumulative germination percentage on the vertical axis versus date. We just monitored when these seeds came up in these flats. And so the species are coded by the treatment. So the legumes that receive the seed coat break are in the light orange. the legumes that were just soaked in water are in the dark orange, and the other two non-legume treatments are in the green. And so from here, what you can see is that for the legumes, under both of these treatments, more than half of the seeds that did germinate did so within the first 10 days of the experiment. By contrast, the non-legume species had overall sort of lower germination percentages, and it took a lot longer. So the legumes seeds germinate quickly, and they germinate synchronously. So we've investigated this early regeneration strategy hypothesis in other experiments. In this experiment, we planted seeds of 8 legume species and compared it to 14 non-legumes under different light and different soil conditions. And so we wanted to know, how does the early growth of legumes compare to non-legumes in response to this environmental variability? And so here, this plots height growth, or height, as a function of week. And the legumes are in orange, and the non-legumes are in green. And the different treatments, or a combination of treatments of light and soil, are symbolized by the different symbols. But what's really important here, you can just sort of squint and see. And that is that at any given age, the legumes have a much higher, they grow very quickly initially, and they maintain that height growth advantage for weeks to months into the wet season. So stepping back and trying to put this into an ecological context, these are monthly rainfall data from Santa Rosa, Costa Rica, for the 80s. And what you can see here is that these are highly seasonal forests. Rainfall is super pulse. There's a very strong wet season that alternates with a very strong dry season. And so we argue that this strategy of germinating very quickly once the rain starts and then growing as fast as you can, if you're a seedling, this is probably a really good strategy to deploy in this environment with very highly seasonal rainfall. Okay, so the take-home message from this part of the talk is that legumes are most abundant in early successional dry forest, if we're thinking about the Americas. Dry forest legumes, they grow quickly, they germinate quickly, and they grow very fast. And we argue that this may be part of a suite of traits that help adapt them to these early successional dry forest landscapes. Okay. So now, sorry. It's so embarrassing to have to remove your mask. Okay, so now I want to focus on dry forest landscapes in the context of climate change, and in particular in the context of drought. And so this is one of the questions that, that keeps me up at night. And it is the question of, will seasonally dry tropical forests be sensitive or resistant to future changes in rainfall regime? And when we first published this paper, I didn't know the answer. I felt very agnostic. I just wanted to know, which is it, right? I didn't have any skin in the game. I just tell me what's going to happen. And so what I want to do now is answer this question using some tree mortality observations we've made. And the particular questions I want to examine are, is drought linked to tree mortality? Which species are vulnerable to drought? Do traits help explain vulnerability to drought? And can we generalize to other forests? And so again, this is the rainfall record. This is annual rainfall from the 80s for Santa Rosa, Costa Rica. And what you can see is that rainfall is highly variable among years. Some years it's over 3 meters. Some years it's less than 1 meter. But this orange year, which was 2015, is notable because annual rainfall that year was the lowest on record. It was an acute drought year. And what you see here highlighted in green are the years for which we have forest dynamics observations. So we can ask this question, is there a link between rainfall, variability in annual rainfall, and tree mortality? So in 2008, I had a PhD student who put in a number of different small forest dynamics plots across gradients of succession and gradients of edaphic characteristics, or soil gradients, in Costa Rica. And we just kept monitoring these plots ever since. So it probably wasn't the best strategy to get a PhD, but now we have like 12 years of data. And each one of these plots you can see up above. And so this figure here, the vertical axis to the left shows the mortality rate, the average mortality rate, or the number of trees, the percentage of trees that die every year in these 18 plots. Annual rainfall in Santa Rosa is on the other vertical axis in blue. And so here what you can see is that, on average, each year in our sort of early to mid-successional forest, 1 to 3% of the trees die each year. By contrast, mortality rates doubled to tripled during this acute drought in 2015. And they remained elevated in 2016, even though rainfall rebounded. And so I would argue, of course, this is correlative data, but I would argue this does give us a strong idea that there is some kind of link between drought and tree mortality. And so our next question was, how are different species affected by this drought? So here, what we've plotted is the mortality rate of 55 different species during the drought. So these data were collected for species for which we had anywhere from 26 to over 200 individuals to estimate this this rate of mortality during the drought. And so what you can see here, and Natalia asked if I needed a pointer, and I said no, but now I'm like, now I do. What you can see here is that there's this huge range of vulnerability in response to drought. So some species lost 35% of the individuals died during this drought. Other species lost no individuals. So some species are very resistant to drought, and some species in this landscape are very, very vulnerable to drought. And then, of course, we wanted to know why. Like, are there traits, are there things we can measure that help us understand this enormous range of vulnerability in response to drought? And so when we started this odyssey, I think, that took about 3 years to figure this out, we've tried all the kind of usual traits, right? The usual suspects. We looked at wood density, which we had databases of. We looked at specific area, specific leaf area, because we had databases for those traits, and they really didn't help us understand the vulnerability across species to drought. And so then we had to drill down deeper and measure things that put me out of my comfort zone, which included physiological or hydraulic traits that might be better linked to tree function during drought. So one of the things we measured was turgor loss point, or the leaf water potential at wilting, or when stomata close. We also invited my friend Tim Brodrib to come and to teach our group how to measure leaf vulnerability to embolism, or P50. It's the leaf water potential at which the leaf suffers 50% embolism. And so Tim has invented this method, or developed this method, that uses a flatbed scanner to track the development of embolism in the leaf as the leaf dries up, and you simultaneously measure water potential, and then you can estimate what water potential the leaf basically died at. So here, let's see, here is it's a matrix of scatter plots. And in this, in these scatter plots, we used each species as one observation. And what this shows in the yellow box is the mortality rate during the drought. So we measured, again, these hydraulic traits of P50, turgor loss point. We calculated an index of hydraulic safety margin. We looked at wood density, specific leaf area, foliar phosphorus, nitrogen, all these other traits, including traits about species distributions under different rainfall niches. And to make a long story short, the variable that was best correlated with the vulnerability or with mortality rates under drought was the hydraulic safety margin. And so here I'm zooming in on this, and this has the different species. Each species is a dot. They're proportional. The size of the dot is proportional to the number of trees that we had to estimate that mortality rate during the drought. And so mortality rate during the drought is on the vertical axis versus our measure of hydraulic safety margin, which we estimated as Turgor loss point minus P50, or the leaf water potential at 50% embolism. And so what you can see here is this strong negative correlation. Species that had a very wide hydraulic safety margin suffered less mortality during the drought than species that had a very narrow hydraulic safety margin. So this is how I think about it, or how I visualize it. This is a figure from a paper Brendan Choate wrote, and so on the horizontal axis here we have an axis of increasing drought stress. When you're measuring Turgor loss point, you're essentially measuring the decrease in stomatal conductance as a function of increasing drought stress. So we can think of that as this blue line here. By contrast, when you're measuring P50, you're measuring this red line here, or the increase in embolism, in xylem embolism or xylem blockage as a function of increasing drought stress. So if we think of a species here, if we think of this as a trait or property of a species, this species here shuts down its stomata far in advance of when it suffers 50% loss of xylem function due to embolism or conductance. Sorry, 50% loss of conductance due to embolism. So really that's what this blue arrow describes, this, a species with a wide hydraulic safety margin. By contrast, we can think of another species that has a very narrow hydraulic safety margin. For this species, indicated here by the dashed lines, this is a species that only shuts down or starts to shut down stomata after it has started to suffer cavitation or embolism due to drought. And so this is a species that when an extreme drought comes along, it dies. So in this study, what we've shown is that there is this strong correlation between vulnerability to drought and hydraulic traits. This study was notable in that we were lucky to have this record of forest dynamics that allowed us to sort of gear up and take advantage of understanding these processes under an acute drought. This study was notable in that, first of all, it was notable in the magnitude of the mortality that we observed. Many people at meetings started calling me like the woman whose trees are dying. And many people I know were jealous of like how much mortality we saw. We were pretty sad when we first started witnessing all the trees die. But I think what has come out of this is, for me, a much better appreciation of how these hydraulic traits are linked to demographic variability in response to global climate change type droughts. But the big question that hit us after kind of spending a couple of years trying to figure out what was going on in this drought was, okay, we think we kind of have a handle on what's going on in this forest in Costa Rica. Does this help us know anything about other tropical dry forests? Can we generalize about what we know in Costa Rica to other forests in the tropics? Another way to ask that question is, what unites and what differentiates tropical dry forests? Okay. So this question became a big focus of the work that we did after the drought work. And what we really wanted to do was to ask the question, to what extent does what we've learned in Costa Rica apply to other places? And so we did this work in collaboration with David Medvigi, who's at Notre Dame University, and his student, Shangtao Zhu. Working with David and Shangtao, who are modelers, what we've tried to do is to develop biologically rich simulation models. And so my team is sort of the data team, and they are the modeling team. And so this schematic here just shows a version of the ecosystem demography model 2, or the ED2 model, that Shangtao has worked to develop plant functional types that are based on hydraulic traits. So this is a simulation model that was sort of based on our data in Costa Rica, and our goals were really to say, hey, can we use this everywhere? Does what we know in Costa Rica apply to other places? So again, what we did was to call all our friends and ask them if they would help us out in their different field sites with this project. Okay, so now I'm going to shift gears. So I've talked a lot about our study sites in Costa Rica and what we've learned there. And again, I want to broaden up the question and ask at a larger geographic scale, does what we've understood there apply to these other sites? And so we called this project Dry 4. If you know Yadvinder Mali, the great Yadvinder Mali, he has this amazing network of forest plots that he calls Rainforest. And we said, well, we're Dry 4 because we only have 4 sites. So what we did was to visit our friends in Mexico, in Puerto Rico, in Colombia, and to really try to generate larger databases of hydraulic traits to understand this variation within and among dry forests. And so this is a figure from Karina Banda and her colleagues in another network called Dry Floor. And what they did was delineate, they used forest inventory data and occurrences of different taxa to delineate the floristic provinces of dry forests in the Americas. And so you can see sort of the different, I think 11 different floristically distinct regions that they identified. And what we did was to sample 4 of these different floristic regions that are indicated by the stars there. Oops. Oh, whoops. That went way too fast. Sorry. So again, what we did was to call up our friends and say, hey, can you guys come? Do you guys want to be involved in this network? We really want to try to we want to dive deep, but do it in a geographically explicit context. So we had colleagues in Colombia, in Mexico, in Puerto Rico, and this is the field team from our site in Costa Rica. So this is Dr. Herman Vargas, who did much of this work for his PhD. He's the newly minted doctor. And so what Herman did was to visit these 4 different sites. And so we have 4 sites we worked in. In each of these sites, we collaborated with our local collaborators to leverage forest inventory plots. And Herman went there and sampled hydraulic traits on the dominant species, or the most abundant species, in each of these different forests. And it took him a long time. He had lots of adventures along the way. So when we started this project, the first thing we did was to try to collate the climate data for these different sites and put all the climate data on one page. And this is that page. So the Colombian sites are in blue, the Costa Rican sites are in green, Mexico in red, and Puerto Rico in yellow. And we looked at a different number, we used a different number of metrics to kind of understand the climate. Annual rainfall is the first panel. The seasonality of that rainfall. So that's a metric that describes whether or not each month has an equal amount of rainfall. So that would be an aseasonal forest, or if all the rainfall is concentrated in one single month. So you can see right here that our site in Costa Rica, which is the green site, has the highest seasonality. It has 6 months of rain that alternate with 6 months where it doesn't rain. Very high seasonality. It has high rainfall, but very high seasonality. We also looked at things like maximum climatic water deficit and dry season length. And you can really contrast the dry forest site in Puerto Rico, Guanica, which has very low rainfall with kind of intermediate seasonality. It doesn't really rain much there, but the rain that does come is pretty unpredictable. So this was a big shock to me, just how many different ways there are to be a dry forest climatically. We also looked at forest structure. So this is just one metric. These are frequency distributions of stems of different sizes. And you can see how much they vary among forests. The Puerto Rico site is dominated by many, many, many very small diameter stems. And this is not a succession effect. This is our oldest forest, but it's dominated by these super, super skinny trees that really don't grow. So this is an example of some of the data that Herman generated. So he went to all of these species. He measured a bunch of hydraulic traits for the different species. And so here, the forests are color-coded again. The species are on the bottom. And this is just one trait. It's turgor loss point. And so with very high values or less negative values, indicating species with low turgor loss point, more less drought resistant, and high values or large negative values indicating drought tolerant species. And what you can see here is that there's variability among species within a forest, and then there's variability among forests. So this is a different way of looking at those data and some of the hydraulic data. This is turgor loss point, leaf vulnerability to embolism, or P50, and hydraulic safety margin. And in this figure, what Herman did was to calculate these community-weighted mean values. So essentially, by linking the species, the value of each one of these traits for each species by the relative abundance of species. And what we can see here is that for the hydraulic safety margin, which again was the variable that was best correlated with mortality rate during extreme droughts in Costa Rica, if I had known this figure 10 years ago, I would have been able to say, hey, on average, this forest in Costa Rica is most vulnerable to drought. It has the lowest, the most narrow hydraulic safety margin, in contrast to this forest in Puerto Rico, which on average, trees in this forest have very wide hydraulic safety margins. And indeed, what we found was that the Costa Rican forest suffered most mortality during this extreme drought, and these other forests really didn't. So in summary here, well, we can't just extrapolate from Guanacaste to everywhere else. But I think we can use our understanding of plant hydraulics, across these gradients of climate and really understand, or use this as a framework to understand which forests are most vulnerable to things like acute drought. Okay, and so last, I'm just going to talk really briefly about some of the soil biogeochemistry we did in these sites. And I think the punchline is pretty much in this picture. And this work was led by Dr. Bonnie Waring. She was a postdoc in my lab, and now she's a senior lecturer at Imperial College in London. And so what we wanted to know in this study was from these same 4 dry forest sites, our little dry forest network, what are the dominant scales of variability in soil biogeochemistry? And we wanted to look at both spatial and temporal variability. And so what we did was sample these same plots, and we sampled the same locations in each plot in the wet season and the dry season. And then we measured pretty much everything you can measure in a Ziploc bag of soil. So we sent everything to my lab in Costa Rica, and we measured soil moisture, particle size distribution or texture, total elements, carbon and nitrogen, microbial stoichiometry, so microbial carbon biomass, microbial carbon, nitrogen, phosphorus biomass. We had a colleague do a phosphorus fractionation We looked at potential net mineralization. We did selective dissolution analyses to look for mineralogy. We looked at extracellular enzyme activity as a measure of microbial function. And we also looked at community composition, microbial community composition through PLFAs. So again, this was just a huge amount of data that Bonnie took to calling Soilmageddon. And so our predictions for what we would find were based on this really cool paper that came out in 1989. And so what these folks did for a site in India was to measure the size of the soil microbial biomass pool in the wet season and the dry season, and metrics or measures of the function of the soil microbial communities, and they looked at nitrogen mineralization. And they found this really cool thing. What they found in this dry forest in India was that the microbial biomass pool was smaller in the dry season compared to the wet season. So if we use this little figure here to indicate the pool of microbial biomass, it was smaller in the wet season, larger in the dry season, but the function changed. So in the wet season, this small microbial biomass pool was turning over very quickly and doing lots of end mineralization. And the sort of dry season had kind of quiescent microbial biomass that was just kind of hanging there, not doing anything until the rains came. So this is what we thought we would find. So this shows soil moisture in the dry season, which is in the stippled bars, and the wet season in the filled solid bars for these 4 different sites. And happily, as we had hoped, soil moisture was higher in the wet season during the dry season. So we're like, we're doing something right. But pretty much nothing else. Everything else was different. This is soil microbial biomass carbon that we got from fumigation extraction. And again, we predicted lower pools of microbial carbon in the wet season compared to the dry season. And that's what we found in Costa Rica, but it's not what we found in Colombia or Mexico or Puerto Rico. So we kind of found every pattern you might expect. Bonnie did this really nice variance decomposition analysis to really try to understand the dominant scales of variability that differentiate these soil observations. So the different factors are indicated by the different colors over there. And in the top panel A, these were things that we only measured on one season. So we didn't expect things like soil sand or clay concentration to vary much seasonally. And what we found for most of these more sort of static properties was that site was the dominant, site or forest was the dominant source of variability. By contrast, in panel B here, anything in white is variance that was explained by seasonality. And luckily, as we expected, soil moisture, a lot of the variability was due to season. Pretty much none of the other variants and anything else we measured did season have a strong impact. And in fact, I think really interestingly, the properties that are indicated in blue here are microbial biomass phosphorus, and then all of the extracellular enzymes that we measured. And for these measurements, the largest source of variability was within plot. And this basically means that, we saw as much variability in a soil sample that was taken over here and 5 meters away as we did find in soil samples that were taken hundreds and hundreds of kilometers apart. Okay, so to sort of wrap this up, this paper by Karina Banda et al. Showed this large floristic diversity among tropical dry forests in the Americas. And I think the implications here are that if you're interested in biodiversity conservation, you can't just conserve biodiversity in one of these regions. If you want to conserve biodiversity in the dry forest, you have to think about continental scale. Our studies here showed that if you want to measure soil biogeochemistry, if you want to model soil biogeochemistry, or incorporate that into a model for dry forests, you can't just extrapolate from one dry forest to another dry forest. And here, is how do we move forward, right? Well, I think one way we can move forward is to think about collecting data across gradients, across climatic gradients, across edaphic gradients, across floristic gradients, and across disturbance gradients, and then really use this approach to understand these dominant scales of variability. Okay, so take-home message from this is that we found high spatial variability in soil biogeochemistry and much less temporal or seasonal variability than we had expected to find. Okay, so I'm wrapping up here. The one thing I want you guys to take away from this is that seasonally dry tropical forests are awesome. They are a totally awesome biome. But they are vulnerable to anthropogenic pressures, including drought. We need a comprehensive understanding or vision of what unites tropical dry forests and what differentiates them. And I think that this kind of approach is really essential for being able to model and understand or forecast the future of these biomes in this rapidly changing world. So with that, what I want to do is really highlight the contributions of Daniel Perez Aviles. He's my lab manager in Costa Rica, and much of this work rests on his shoulders. So then I want to thank my collaborators and the funding sources that have made this work possible. And as I promised, if you guys are still awake, I will read a bedtime story to you. This is a story that I wrote when this is embarrassing, in 1995 when I was working at a field station in Costa Rica. And it's a wet forest story, so it's a rainforest story. But anyway, here we go. This is called Night and Day in the Rainforest. The sun was setting in a rainforest in Costa Rica. Parrots were squawking as they flew home to sleep for the night. A young coati named Pisotito was not sleepy, as he explained to his mother. Coatis sleep in the trees at night. After his family was asleep, Pisotito climbed down the tree to explore the forest. He was surprised to meet an animal he had never seen before. Her name was Paquita, and she was a paca. Pacas are nocturnal. They sleep during the day and are awake at night. Paquita took Pisotito to meet some of her friends in the frog swamp. They silently watched the jaguar from behind the tree. Day was breaking in the rainforest. Some animals like the kinkajou were getting ready to sleep, and some like the tinamou and the anteater were just getting up. Pisotito brought Paquita home to meet his mother. She suggested that Pisotito introduce Paquita to some of his friends. Pisotito's friends were diurnal animals. They were awake during the day, like the turtle warming herself in the sun by the riverbank. When the sun was high in the sky, they met a troop of spider monkeys and an eyelash viper. As day was turning into night, they said goodbye. They both thought about how the forest is full of interesting animals all day and all night long. And so what's the point of this? Other than I really like to draw coatis. I think the point of this is that as scientists, we can take our passion for what we study and communicate it to larger audiences. And we can do this in a number of different ways. For me, I like to draw. But for whatever you do, take what you're passionate about and let other people know why. And I think that is one way forward out of some of these dark days that we've been experiencing this last decade. Okay, so muchas gracias. I'll stop talking and I'll say thank you. And if you have any questions, I will try to answer them. Thank you. Yes? Yeah. Yeah, that's a really good question. Yes. So the question was, how much of a role might tropical dry forest play in CO2 sequestration? Yeah. So there's sort of twofold answer. One is that these forests tend to have species that are, that might grow slower than a rainforest, but they have very high wood density. So, a mature tropical dry forest likely has high carbon stocks. It might not be, a super tall, it might not be equivalent to a dipterocarp forest, say in Asia, but they can have high carbon densities. The other answer to that is that the tropical dry forest biome has been much more impacted by land use change than the tropical rainforest. And so much of, the forest that I work in, which is the Mesoamerican dry forest, which used to extend from Mexico all the way down past Panama, they estimate that 2% of the original extent of that forest is left. And so the other answer to that question is that because these forests have been so degraded, they've been so transformed by land use change, there is a large potential for regeneration and reforestation. Natural forest regeneration, we look at succession, and that is really a viable, if it's done in the right way, I think that's a very viable nature-based climate solution. So yes, both of them. Anything else? Yeah. Oh, sorry, one in the back, and then, yeah. I think that's Chris. Yeah. Uh-huh. Yeah. Yeah, no, so that's a really good question. Like, so what, and there's kind of like the philosophical angle, like what is a trait? And then there's sort of the practical implications of like how plastic is it? And there's this very, what is the intraspecific variability? Yeah, so there are some anatomical features that are correlated with these kind of hydraulic traits. Like wood density is somewhat correlated with it. Xylem, this, vessel size, that sort of thing. Vessel size can be correlated with this, with these hydraulic, characteristics. So yes, I guess, yes. I mean, obviously, like, yeah, yes, I guess. Yes, they kind of have to be, right? Yeah. Yes, right on. Hopefully I did answer your question in some roundabout way. There are anatomical features, but knowing these, I think that having a sort of flux-based, or these are more, that these are better associated with these processes, I think, than the more static. Like wood density didn't help us explain this. Yeah. Mm-hmm.
Thanks, Jennifer. That was a very nice talk and story. I find similar results when I look at roots in terms of their variability. So roots of the same tree differ more than roots of trees of a different species along a super long radius.
Yeah.
And my explanation is always that the soil is very heterogeneous and that influences the variation in root traits. But I always struggle with how to then continue, right? And how important do you think that this super large variation in soil biogeochemistry is for tropical dry forests, or forests in general?
Yeah. So that that's a good question too. The picture that I showed with all of the different colors of soil is so nice because that those colors also reflect different properties, right? Texture, mineralogy, stuff like that. There's different ways that soils affect plants And then, of course, there's different ways that plants affect soils. And so I think it's, so this is one of these classic kind of you need to know the scale questions. But our work in Costa Rica, I could have shown you a picture of our soils in this small region we work in Costa Rica that has as much variability in that region as we see across these forests that we looked at in different sites. And we've been looking at things like how that variability in soils affects species distribution, right? And so we definitely see species that, in our sites in Costa Rica, one thing that's pretty unusual about them is that we have some very low fertility soils that are dominated by a species of quercus, an oak species that's ectomycorrhizal. So some of the soil variability is important because it's acting as a filter, as an environmental filter for species at that very coarse scale, right? We see that too with legumes, like where across a soil fertility gradient, within the same climate, if you're looking at a soil fertility gradient, we see legumes in some places and more legumes here and fewer legumes there. So I think, how do you grapple with that sort of question? I think one way forward, I think, are shade house studies. I mean, I think that those are pretty interesting for looking at this, for looking at what root traits or root characteristics are properties of species versus properties of the environment. Yeah, and so a student of mine just pre-COVID finished this. She grew N-fixers and non-fixers in a shade house under different phosphorus conditions, and then looked at phosphatase activity, specific root length. She really tried to look at all these different root traits that are morphological, that are functional. And what she found was that a lot of the variability actually was at this legume non-legume split, or at the species, just across a phosphorus gradient. So thinking about those dimensions of variability, how much variability do you find, and partitioning that into what's due to a species, what's due to environment. I think those kind of experiments are really good ways to do it. That's what I would say.
So sort of try to quantify the contribution of that.
Of that plasticity, right? Right, exactly. Yeah. So, yeah, question in the back. Yes, yeah. Uh-huh.
Are there the soil biogeochemistry or the compositional variation follow bedrock geology?
Yeah. Yes, absolutely. So that soil variability, and so some of the variables that we measured in that study were really linked to, that variation in soil was very much inherited from parent material. Yeah, absolutely. And we could see that with the forests in Colombia. They were inner Andean valleys, and they just had 4 times more phosphorus than any of the other sites. Yeah, so definitely. So like a Hans Jenny approach of thinking about soil forming factors or ecosystem state factors, where you think of climate, topography, parent material, organisms, weathering time, that all very much informs, I think, how we think about these things. So definitely, yeah. Anything else? Yeah, okay,
Thank you for the talk. Can you repeat, how you measure,
Yeah, that's a great question, because how do you know when trees die? Yeah, that's a great question. It's not when a cow dies, you know it, right? You're like, that cow is dead. But when a tree dies, how do you know it? So that's a good question. So we noticed that the trees were dying during this drought, and a lot of them had dropped their leaves, which is very, very unusual. And then you could go and you could take a twig and just kind of break it, and they were dried out, so they looked dead. And so anything that had that look during the drought, we said, okay, that's dead. And then we went back in 2016 to make sure that everything we thought was dead was really, really dead. And so luckily, or I mean, sadly, like however you think of it, sadly or luckily, everything we thought was dead in 2015, you know, was mostly dead, right? Like, yeah, it's like that scene in Princess Bride when they're like, are you mostly dead or totally dead? Most of those trees that we thought had died in 2015 were still dead in 2016. And so, yeah, and then we knew, hey, we know how to tell a dead tree from a live one. And with lianas, it was super easy because, you know, my lab manager said to me, he's like, these lianas are dying. And I was like, how do you know? And he just took one, he snaps it. Boom, okay, that's dead. Yeah, so, I mean, we used this criterion of like, were they dropping leaves? And then when we went back the next year, nothing had leaves. Yeah, good question though. Anything else? I think that's it.
Well, let's thank Jenny.
Thank you, guys. Thanks so much.